My name is Hilal Demir. Welcome to my PhD project, Cross-Protection of Allergen Immunotherapy-Induced Antibodies to Related Allergens requires a high degree of structural identity. In our previous study, we saw that sublingual immunotherapy with two homologous allergens, namely recombinant BATW1 and recombinant MALI1, induced allergen-specific IgG1 and IgG4 antibodies. However, recombinant MALI1 induced IgG antibodies showed a high IgE blocking capacity to MALI1 with an improvement in their apple allergy, whereas recombinant BATW1 induced IgG antibodies showed a limited IgE cross-blocking activity to MALI1 and no effect on their apple allergy. This was surprising because BATW1 and MALI1 share an amino acid sequence identity of 56%. Moreover, IgG1 was identified as the major IgE blocker. Therefore, we used this study to answer the research question if the molecular similarity of allergens matters for the cross-blocking capacity of the allergen immunotherapy-induced IgG antibodies. For this, we then chose other BATW1 homologous food allergens such as PruF1 from cherry, Cori1 from hazelnut, and Dausi1 from carrot. And as you can see, PruF1 has a really high amino acid sequence identity to MALI1, whereas Cori1 and Dausi1 have a similarly high amino acid sequence identity to BATW1 and MALI1. First, we quantified the IgG levels induced after treatment, and we saw an increase from pre to post treatment for recombinant MALI1 induced IgG antibodies to MALI1, PruF1, and Cori1, and for recombinant BATW1 induced antibodies to all allergens except Dausi1. To characterize the IgE blocking capacity of the induced IgG antibodies, we used basophil activation tests where we calculated the inhibition of pre to post treatment. Recombinant MALI1 induced IgG antibodies showed a significantly higher inhibition of basophil activation when stimulated with MALI1 or PruF1, whereas recombinant BATW1 induced IgG antibodies showed a significantly higher inhibition of basophil activation when stimulated with BATW1. To answer the differences in blocking capacity, we wanted to see if the induced IgG antibody showed a different binding affinity. For this, we used two different experimental setups. One of them was the disruption of allergen antibody complexes with acidic conditions. This is an ELISA protocol where we first coat with the allergen, then incubate with the post sample containing the IgG1 antibodies that we are interested in, then incubating with an acidic buffer, removing the low affinity IgG1 antibodies, and then detecting the remaining IgG1 antibodies. The second method is a competition ELISA between binding to MALI1 and the competitor allergens. For this, we pre-incubate the post sample with a competitor allergen, then add it to a plate where MALI1 is already immobilized, where only the IgG1 antibodies that did not bind the competitor allergen can bind to MALI1 and are then detected. And to simplify the results, we saw that the IgG1 antibodies induced after MALI1 treatment showed a stronger and higher binding to MALI1 and PruF1. This could possibly be the case due to differences in epitope recognition. After comparing the surface structures of all allergens, we noted that PruF1 has a high identical surface area with MALI1 and not so high with BATW1. When we had a closer look on PruF1 molecularly, we saw that there were identical surface area patches only shared between PruF1 and MALI1 and absent on BATW1, indicated in the green color. These patches could possibly contain epitopes that are recognized by the IgG antibodies induced after MALI1 treatment, but not after BATW1 treatment. Therefore, we concluded that high structural identity of allergens is required to induce cross-blocking antibodies.